Hey, it's Eddie from Menu for Molecules for Life here at the 2016 Bio-Photonics West Expo in San Francisco, California. This is the largest medical bio-optics and biophotonics event worldwide. So come with me, step through the looking glass, let's see who's here and what they have to show us.
I'm Mary Harper. I'm VP of Business Development at Edeluma Inc. in Carlsbad, California. We have novel, high-resolution digital inverted microscopes that we make that have a variety of different features that make them very useful for scientists in all kinds of companies or in academics. We also OEM. We have a number of agreements where we're using components or even the whole lumiscope that we sell. So our microscopes are called lumiscopes, but they are actual microscopes. So let's look at one of our manual ones. This is our lumiscope 620, and it does 3-color fluorescence. It is inverted. The optics are very simple. If you want to show the optics over here. So there's a camera at the bottom, then there's the light block, there's a photo tube, and then there's an objective. So by having this so simplified, and it's also solid state, we get very high resolution, as good as what you would get on a Zeiss or Nikon or other type of upright microscope. We retain about a majority of the light as opposed to losing it, which you get on a traditional microscope. So what we're showing here is a typical 3-color fluorescent image, the red, blue, and green, and again, high resolution that you can see there. Our microscopes are also very compact, so this goes into an incubator or into a hood very readily. It's a big advantage for us because they go in and out of incubators just fine for live cell imaging. They're very versatile. You can use just about any labware. This particular one connects to the computer, and it's powered and controlled by the computer only, so you don't need a separate power cord. So you can use this out in the field or other places where maybe you don't have an electrical power cord. It also takes time-lapse. It's one of the main applications that we have. It also does live video. And again, we are adapting this for various companies. These components can go into a different instrument if you need such an imaging capability and in high resolution. Now let's step over to our automated version, which is our 720, which we're just introducing now. Same optics as the 620, 3 colors. We are working on more than that, other sorts of features. This one has an automatic stage, very high precision, so it will do microplates up to 1536, which again is a feature that other companies don't necessarily have. Autofocus, and it uses an algorithm, so it's image-based, and all of our lumiscopes are run by our own software that comes with it called LumaView. So you do all the experimental parts of image capture through that. And in this particular one, again, we're doing 3-color imaging. And if you look at the monitor right now, it's autofocusing in the blue channel. And now it's in the green channel, so it's focusing, and then it will take a picture, and then it focuses in the red. So because the LEDs and the filters, it's a people set, are fixed, it's solid state, the 3 different channels are exactly superimposed on each other. We can also do bright field and phase contrast. So we offer a lot of different options, and please check out edeluma.com, and we'd love to hear from you. Thanks. Bye.
Hi, my name is Xiaoxi Zhen. I work for Kim Labs. So we make ultra-fast laser technology. This is our new technology. We call it Zeus. So we basically convert IR light using this device to soft X-ray machine. So this device can actually produce 4 nanometer EUV light. So this light can be used for nano imaging, especially very useful for biological applications. This wavelength is called near 4 nanometer machine. This light source can be used for this wavelength actually is called water window. The water window is good for detecting oxygen and carbon. So with this light source, you can imaging not beyond the cellular level into atomic level. So this is our EUV generation device. So this red box actually is takes a high power femtosecond laser and generate extreme ultraviolet. And this module, after this source, there is several, lots of modules to manage the EUV beam, because the EUV beam only works in the vacuum. So to deliver this laser beam requires a lot of requires a lot of vacuum technology and special optics. So look like so the first module is used to monitor the laser beam coming through the fiber, and then the second module here is to split, because we use IR beam to generate EUV. We use this module to split the IR and the EUV. And the third module here is to block the IR beam completely. And this one is actually a focusing optics, so we can focus the EUV light onto the biological samples to do imaging. And this is again a filter to further remove the IR background. And then the final module is actually the module to spread a spectrum, because the laser light coming out from here actually has a lot of colors. So this module selects only a single color and to feed on the biological sample. And in the meantime, this one can also use to tune the colors. So you can use different colors to illuminate your sample.
Ben Barbieri, ISS. FRET is an acronym for Foster Resonance Energy Transfer. It measures if two molecules get closer together. The idea is that if we have molecules A and B, and if we target one molecule absorbs photons, it goes to the excited state. If molecule B is close enough to molecule A, close enough means below 10 nanometers, the energy gets transferred from molecule A to molecule B. And in the process, we can measure this process and determine if the two molecules are close enough, and by the efficiency, even determine the distance between these two molecules.
Hello, I'm Uwe Ottmann from a company called PicoQuant. We're building confocal microscopes for film and FCS. It's basically a machine which has the versatile property of the dye in 2 dimensions, and you can also measure correlation times of molecules. It's basically a machine which works on the single molecule level. You don't look at the ensemble of versus molecule, but just as protein by protein or molecule by molecule. That enables you to look at the individual properties of the actual dye. It's basically a confocal microscope, but it's also time resolved, and also you can get an image. Thank you.
Hello, my name is Bob Crescent. I'm president of KS Technologies. We're a design house with Texas Instruments, and we have taken the Texas Instruments DLP technology, which is used in projectors, and crafted a near-infrared spectroscopy device. So over here on the demonstration station, what you'll see is the device that we have created. So we've productized a DLP-based spectrometer. It goes from 900 to 1700 nanometers with about 6 nanometers of resolution. We have 4 of them that we're showing here that are Wi-Fi connected to a Dell 5000 series IoT gateway, and then the IoT gateway is connected to the cloud. So what we can do is use spectroscopy for things like materials identification, or even looking at chemical concentrations. So here's an example of a sample. Now I'll go ahead and put it onto this fixture, and I'll initiate a scan. And what we're doing now is taking this and comparing this scan versus a reference database that we have created, and we're seeing if there's a match. So over here on the display, we're performing the analysis. We have our reference scan on the left, the actual scan that we just took here on the right, and we've determined that there is no match. So during research for your studies, this can be a great, inexpensive tool to look at the molecular concentration of almost anything. If you have any questions, you can reach out to us on our website, which is kstechnologies.com. Thanks for your time.
Hi, I'm Scott Rutter with Innovative Photonic Solutions, and I'm here to tell you a little bit about Raman spectroscopy and some of the products we've got that specifically help you detect different kinds of things chemically.
Exactly.
So Raman spectroscopy is an analytical chemistry technique that allows you to detect what a molecule is made up of. So you can take a look at how the bonds are being configured and how the whole molecule is actually being stretched. So it can tell you polymorphs, different formulations of the same object, and crystal structures as well. What's unique to our products is that we focus specifically on providing really innovative solutions that provide really, really high throughput, so effectively optimizing the system as opposed to just putting systems together willy-nilly. So what we've got right here is an innovative new product. It's called an integrated Raman probe. What we've figured out is that the current state of the art, if you will, is to buy a spectrometer from one company, a probe from a different company, and a laser from yet a different company. And by doing that, unfortunately, these companies don't necessarily talk to one another real well, and there's inefficiencies in the system when it's combined. And that inefficiency leads to light loss, and light in this particular case is something you don't want to lose. Raman spectroscopy is a 1 in a million event.